Hello, welcome to the first lecture in Chapter 12. Chapter 12 is going to be covering DNA structure and DNA replication, processes, and a little bit of mutations, what happens with DNA when it gets mutated. So this first mini lesson is going to talk specifically just about the structure of DNA, and then I'll do some other mini lessons on setting up and getting ready and going over the process of replication. So you had a little bit of introduction to DNA back in chapter 3 as one of our major organic macromolecules, right? So nucleic acids. DNA is probably the most common of our nucleic acids. The other ones we have is RNA and nucleotide derivatives like ATP, NADH, FADH2. We saw those in the metabolism chapter. So what I wanted to do for this little mini lesson is just talk specifically about the actual chemical and molecular structure of DNA. A little bit of history before we get there. You may have heard of the guys Watson and Crick, shown here in this picture as they're pointing to a large three-dimensional model of their structure of DNA. Prior to Watson and Crick publishing their paper, I think in April of 1952, so the people knew about nucleic acids. They could chemically analyze DNA, but they didn't know that DNA was a molecule of heredity for quite some time. So people were hypothesizing that could have been proteins or carbohydrates or one of those other large macromolecules, but certainly not nucleic acids. It was just 4 letters, A's, T's, C's, and G's. How could such a simple molecule code for all of the instructions of every living thing? Once it was determined that DNA was the molecule of heredity, and you can read that in the kind of some of the historical experiments in chapter 12, then the race was on to try to find and kind of determine the structure of this DNA. What did it look like, and how did it allow for this inheritance of traits from one generation to the next, since it is a relatively simple molecule? So they won this race. Watson and Crick won the race by publishing their paper in the journal Nature, and like I said, in 1952. 52 or 53, I don't know, one of those years. But I also have pictured Rosalind Franklin here, who she specialized in X-ray crystallography, diffraction crystallography, which is this picture here. So the idea is this is the outer edge of the DNA. It would be like if you're taking a double helix DNA and like looking at it down the tube, if you will. So the outer edge is here, and then this X is representing the rungs of the ladder as it's kind of going through this helix. So her picture of DNA in this particular way, like this X-ray picture, allowed Watson and Crick to be able to get the measurements they needed to finalize their idea of the structure of DNA. So there's some controversy as to whether they got that information by consent. Did she share that information freely, or was it acquired in not so free ways? So there's some controversy there. But she died before the Nobel Prize. They won the Nobel Prize, by the way, for their contribution to determining DNA structure, but she died of cancer because she dealt with X-rays and radioactive things, and they don't award Nobel Prizes after people have died. So I do want to always put her out there as she was a big part of their discovery, even though she may not have been officially awarded that Nobel Prize. So when we take a look at the nucleotide structure, again, we've seen this before, so we can review some of these things. The nucleic acid is a very large molecule. It is a polymer, right, of many smaller units, and the smaller units are the nucleotides. So I'm going to circle one nucleotide right here. So a nucleotide is made up of a phosphate group, a sugar, and a base. So here's the phosphate group, here's the sugar, and then here's the base. So it's got those 3 parts. And then when you start hooking these together, you get a repeating pattern of a sugar, a phosphate, a sugar, a phosphate, a sugar, a phosphate, phosphate, a sugar, a phosphate. So this is what we typically call the sugar phosphate backbone, which is on either side. I'm just showing you one side here, with the bases kind of sticking out towards the middle. All right, so this is our sugar phosphate backbone, and the nucleic or the nitrogenous bases, the A's, T's, C's, or G's, are kind of pointing towards the middle of this double helix, all right? Another really important thing to identify here, I'm going to get rid of all my markings, is the numbers of primes, particularly the 5' and the 3'. Okay? So this carbon right here and this carbon right here. The primes are ways for organic chemists to keep track of the carbons in a particular molecule. So in sugar like deoxyribose, which is highlighted kind of pinkish here, this first carbon is called the 1' carbon because it's kind of clockwise to the oxygen. So here's our oxygen. And then that first carbon is called 1'. And then we kind of go around clockwise. Here's 2', 3', 4', 5'. So it's just a way to name tag those carbons. When we are taking a look at how these nucleotides connect to each other, it is the 3' carbon and the 5' carbon that are involved in this bonding between your adjacent nucleotides. So if we come down here to look at the bottom, let me get to my pen here. We'll do red. So this 3', right, is an open 3' carbon, this one right here. It's not bonded to anything. So when we take a look at a big long piece of DNA, something like this, if this end is a 3', we can just label it like that. All that means is the unattached 3' carbon. It's just got an OH sticking off of it. There's nothing else stuck to it. Then if we go up to the top, right here, that's where the phosphate group is bound to that 5' carbon. This would be our 5' end. Okay. So you're going to see that a lot, especially when we get into the replication, because the position of the 3' and the 5' carbon is really important to how the enzymes work to be able to actually do DNA replication. So when we talk about the 3' end and the 5' end, all we're referring to is the unattached ends of the 3' and the 5' carbon. 1, 2, and 4 don't play any part in the bonding between these adjacent nucleotides. So we'll be talking about that more, but this is where those numbers come from. Okay. Then we take a look at those partnerships between the bases. So if this was our, double helix, so here's our sugar phosphate backbone, then the bases sticking off, we have an A matching with a T, and we have a G matching with a C. So you might remember this from chapter 3, is A's pair with T's and G's pair with C's. This is what's called Chargaff's rule or the base pair rule. And what he determined as he looked at a whole bunch of different kinds of DNA, right? So here's all the different kinds of DNA: E. coli, yeast, sea urchin sperm, herring sperm, human liver, and corn. So bacteria to fungi, to animals, to plants. If we take a look at the ratios between A's and T's and C's and G's, they're almost always right around 1. So however much A's you have about the same amount of T's. However many G's you have about the same amount of C's. So in human liver, we can see it's almost identical. The A's and T's are identical. The G's and C's are really close to being identical. So those are Chargaff's rules. And that's really important when it comes to replication, because if you know 1/2 of your DNA strand, okay, let's say you only know 1/2, would you be able to determine what the complementary or other half is based on just knowing 1/2? And the answer is yes, you can, because wherever you have a T, you have to have an A. That's Chargaff's rules. C, G, G, T. Okay? So what that leads us into is when we get ready to replicate the DNA, all we have to do is separate each half and then copy each of those halves, and we can get an identical strands of DNA. And that's exactly what happens with some, a little bit more complex terminology and processes, but that's really the basics of it. You have your DNA split, each side serves as a template, and you get 2 identical strands of DNA. Now, the thing that holds the 2 halves of the DNA molecule together are hydrogen bonds. And that's one of the reasons why A's and T's go together and C's and G's, is because the molecular shape of the adenine and the thymine allow for 2 hydrogen bonds to attract them together, whereas guanine and cytosine, their molecular shape allows them to have 3 hydrogen bonds holding them together. So if you had a thymine, thymine and a guanine, it wouldn't work. You wouldn't have the molecular opposite for them to be able to match up and hold together. All right, so we've got our nucleotides are held together at the 3' and 5' sugar phosphate backbone. We have the bases kind of protruding internally towards the inside of the sugar phosphate backbone, and they pair up A's with T's, C's with G's, and they're held together by hydrogen bonds. Okay, so here's just another more colorful look of these nucleotides and how they are put in this double helix. So we're showing the hydrogen bonds between the A's and T's and between the C's and G's. But I also wanted you to see the arrangement or the alignment of these 2 halves of the DNA, this double helix. So if we look at the sugar, it kind of is pointing like this, and the oxygen would be right here. And this would be our 1', 2', 3', 4', and then the 5' up here. So it is this 3' end that is unexposed, so we call that the 3' end. Now, if we look at that position, we can almost kind of go like this, and that sugar, oops, draw it a little bit better, is in this orientation, okay? Kind of with the point pointing up. That's where the oxygen would be, right here. Now look at the other side. Okay? It's like it's upside down. We can see it a little bit better over here on this bigger picture, with the oxygen here. And so we got to turn ourselves upside down, but this is the 1, 2, 3, 4, 5' end. So when we take a look at this part of the sugar that is exposed down here, that's going to be the 5' carbon where the phosphate group would be attached. It's not shown here, but it'll be a phosphate group here, the PO4. That's going to be on the 5' end. And if we take a look, that is kind of oriented this way with the oxygen pointing downwards. They're opposite each other. We have a special word for this. It is called anti-parallel. So our DNA strands run anti-parallel to each other. So one set of nucleotides are kind of facing this way with the oxygen pointing up on the sugar, and its complementary strand is pointing, the sugar, oxygen sugars are kind of pointing in the opposite direction. So this is what we mean by anti-parallel. And then lastly, this is just some different types of visual representations of DNA in almost like a 3-dimensional form. So again, we can see our base pairs with the A's and T's, C's and G's, the two hydrogen bonds versus 3 hydrogen bonds. And so kind of a space filling model, and then even a more space filling model over here. I'm not so concerned about knowing the major grooves and the minor grooves in these measurements. It's just kind of cool to see the DNA molecule with these atoms. So, right, it's an organic molecule, oxygen, tongue-tied, hydrogen, oxygen, nitrogen, carbon, and some phosphorus. So DNA has those 4 major atoms that are found in 96% of all organic molecules, and it has the phosphorus as well, and some nitrogen in the bases. Okay, so we see nitrogen, phosphorus. Oh, the nitrogen is part of the honk, right? Carbon, oxygen, hydrogen, nitrogen, and some phosphorus. So this is what the DNA molecule looks like. So before you move on to the getting set up and preparing for the DNA replication, make sure you're comfortable with those components of a nucleotide, the sugar, the phosphate, the base. Make sure you're comfortable with recognizing what the 3' and 5' sides are, because that's going to come into play when we get to the replication pieces. All right, I'll see you next time.